So here phase A will be considered as the phase that is present in the majority, and phase B will be considered that is being present in the limited amount. So that's why the distribution of phases is there. So if you have any doubt in this question, you can ask. If not, then we'll move on to the next question. So the partition coefficient, or Kd, depends upon the physical properties of the molecule or the chemical properties of the analyte, or both physical and chemical properties, or is independent of physical or chemical properties of the analyte. Can anybody answer? Okay, someone said option number 3. Any more answers? Yes. So the correct answer is option number 3. So whenever we are studying the partition coefficient of a particular analyte, so in that case, the for example, we have two phases, that is the stationary phase and the mobile phase. So in this particular context, we'll see whether the analyte has uh, attraction towards the stationary phase or the mobile phase. Okay, so in that case, we consider it's the physical properties. For example, charge. Okay, then if it has some hydrophobic patches on it, or it can be dissolved in a polar solvent. Okay, that represents its chemical properties. So the partition coefficient depends upon both of these properties, whether the particular analyte would be attracted towards the stationary phase or the mobile phase. So that particular stationary phase can be used for its purification as well as the mobile phase. Okay. So any doubts in this question?
No, sir.
Okay. Let's move forward. So the question is, given below are the partition coefficient of various compounds. So there are four compounds with different partition coefficients. So A, B, C, D, E, A, B, C, D, with which have a partition coefficient of 5, 2, 10, and 7 respectively. Mixture of these compounds is passed through a chromatography column. What will be the order of elution of the individual compounds? Can anybody guess? Okay. Any more answers?
One, sir.
Okay. So yes, the correct answer is option 1. So what happens in case of partition coefficient? What we consider is how it is attracted towards the mobile phase. So in this case, if the partition coefficient of the analyte that we have is attracted towards the mobile phase, in that case the Kd of the molecule will be less. Okay, because in that case the analyte would be quickly passed on to the mobile phase when we'll purify it. Okay, so in the chromatography column, when we'll run different compounds whose Kd would be less, will be eluted first. So that means the first compound that we'll see will be having the Kd the lowest, and it will be more attracted or more dissolved towards the mobile phase. Okay, and less attracted towards the stationary phase. So this is the concept. The lower the Kd, the more attracted it is towards the mobile phase. Okay, so here the compound B has the least Kd. So it will be eluted first. Then comes the second Kd is option compound A, that is A. Then the 7, that is compound D, and then Kd equal to 10, that is compound C. So this will be the correct order. So that is option number 1. And that means the particular compound will be staying least in the column. Okay, so that's why when the mobile phase will be passed through the column, the compound that have the least Kd will be eluted first. Okay, so any doubts in this question?
No, sir.
Okay, so let's move forward. So the ability of chromatography column to separate two analyte peaks from one another is called distribution, partition, separation, or resolution. Can anybody answer? Okay, resolution.
Sir, 4.
Okay, so yes, so resolution basically means whenever we have to separate any two things. Okay, so in particular case of chromatography, if we can see that two peaks can be resolved further, that means these are the two proteins that can be separated easily from that particular chromatography. In this case we say that the resolution of this particular chromatography is good. Okay, and if there are combined peaks, that means this the resolution of these two proteins is poor. Okay, so the difference between two separate peaks, if we can differentiate between two separate peaks easily, that means the resolution will be good. Okay, so here is the explanation of resolution. And we generally detect resolution in terms of the time that the analyte takes and the width of the peak that we observe. Okay, so there here we injected the sample in the column, and this is the baseline, and then the first protein that we observe is at this point. Okay, then the second peak that we observe is this one, that is A. Okay, here we this is the baseline. That means the first standard or kind of line that will allow us to see whether the chromatography column has been run successfully or not. Okay, then after that we see the separate lines of different proteins that have been purified from that particular chromatography. Okay, so here we can see that at retention time of 1, the A compound has been eluted out. So that means it has been eluted out first. Then comes the B, and then here the C and D. So here, as you can see, that the resolution depends upon the retention time as well as the width of the peak. Okay, so this is the average width of the peak that we see here. So this peak width will be uh, denoted by W. Okay, so here the retention time, minimum retention time means that the KD value will be less. Okay, so this is the formula for resolution, in which it depends upon the retention time of the particular analyte in the column as well as the width, average width of the distribution peak that we observe here. And this particular graph is called as a chromatogram. That is the difference between uh, the graph between absorbance as well as the time in which the different proteins are eluted out. Okay, so this is the particular explanation about resolution that we study in a chromatography column. So any doubts in this? If not, then we'll move on to the next question. So in column chromatography, the number of distribution planes is directly proportional to dash and inversely proportional to dash. So what can be the correct option out of these four?
Sir, 4.
Four. Okay. Anybody else? So yes, the correct answer is option number 4. That is, height of the column is directly proportional to the height of the column and inversely proportional to the height of the distribution plane. So what happens basically? The resolution that we say depends directly on the number of distribution events. So these events are basically based upon the number of particles that are attracted towards the mobile phase. Okay, that means that the number of particles that are being attracted towards the mobile phase will be separated easily. Okay, so here the number of distribution events is now that we denoted as by n will be directly proportional to the length of the column or the height of the column, and inversely proportional to the height of the distribution plane, that is this particular graph. So this is the height of the column. Okay, so sorry, height of the distribution plane. So this is the formula. Now in this particular case, if the number of distribution planes are high, that means the particle will travel less time in the column. Sorry, the particle will travel more time in the column, and the distance between the peaks would be more. Okay, so that means the retention time of the particle will be more because it will travel more time in the column, and this would lead to the distance between the two peaks. Okay, such that increasing the resolution of the chromatography. And secondly, if the peak width is sharp, that means if this kind of peak width is observed, that this is a broad peak width. And if the peak width is sharp, that means it has separated easily, and this will also increase the resolution. So that's why resolution is directly proportional to the distribution events in that particular chromatography column. Okay, so now if the retention time is high, that means the difference between the peaks is more, and if the if the number of distribution plates is high, that also means that the peak height can also be more. Okay, so here as we can see that relative distribution in the column, here it's n is equal to 1000, that means this is resolution is good. Okay, because here the length of the column would be more as compared to the height of the height of the distribution plane. As compared to the second part that has n is equal to 100, and the last one where n is equal to 10. So here as you can see the resolution is minimum. Okay, so this is the meaning of the number of distribution planes and how it depends upon the resolution of the particular chromatography column. Okay, so any doubts in this particular
No,
part?
sir.
Okay, so if not, then we'll move on to the next question. The increase in height of the distribution plane will result in increase in number of distribution planes, no change in the resolution, decrease in resolution, or increase in resolution of the column.
Sir, number four.
Okay, any more answers? Just now we studied the formula in the last question itself. So please try to answer this. Okay. So yes, as we just studied that the number of distribution planes is directly proportional to the height of the column and inversely proportional to the height of the distribution plane. So here height has been increased of the distribution plane. So the number of distribution events will go down. So the first option is wrong. Now the number of distribution events is directly proportional to the resolution of the column. So, if the number of distribution events are going down, that means the resolution is also decreasing. Okay, so the correct option is option number 3. That means with increase in the height of the distribution plane, the resolution of the column would decrease. Okay, so any doubts in this question?
No, sir.
Okay, then we'll move on to the next question. In ion exchange chromatography, the separation of analyzed is based on the hydrophobicity, surface area, charge, or size of the analyte. Can anybody guess? Yes. So, as the name itself indicates, it's ion exchange. That means the property that is involved for the separation of the particular analyte is charge. So, the correct answer is option number 3. Okay. So, let's move forward. In which type of chromatography, the interaction between the matrix and analyte depends upon the isoelectric point. Size exclusion, ion exchange, affinity, or hydrophobic interaction chromatography. Can anybody guess? Okay. Anybody else? Okay. So first, tell me what is isoelectric point. We have discussed in the last lecture. So isoelectric point is the pH at which net charge on the protein is zero. Okay, so this isoelectric point will help us deduce when this particular protein or analyte would have a positive charge or a negative charge, right? So that means if anything related to charge is there, then the chromatography that we use is ion exchange chromatography only. Okay, so everybody understood, or shall I explain again? So whenever we see isoelectric point, okay. So whenever we see isoelectric point, that means we know when the protein can have a positive charge or the negative charge. Okay. So on that basis, we'll use ion exchange chromatography to separate that particular protein. Okay. So we'll move on to the next question. So in an anion exchange chromatography, the column is packed with positively charged beads, negatively charged beads, or negatively charged protein binds to positively charged columns, or both positive and negative charged beads are present on the column. What can be the correct answer? So, by the way, first I would like to know what is anion and what is cation, because I know people have really lot of confusion regarding this. So, what is anion first? Is it a negative charge or a positive charge?
Negative charge.
Okay, and cation is the opposite, that is positive charge. And what is cathode and anode? Is cathode a positive charge electrode or a negative charge electrode? Okay, so please check this and let me know in the next meeting. Okay, here, if you
Cathode
know,
is negative charge.
sorry, cathode is negative charge.
Yes, sir.
Okay, and anode is positive charge. Okay,
Yes.
so how to remember this? If anion is negative, okay, it will attract towards anode. So that means the negative ion will attract towards the positive charge. So this crisscross relation you remember, then you will remember what is anode, what is anion, and what is cathode, and what is cation. Okay, so this is a simple trick to remember this, because in the real life scenario also the people also get confused regarding these terms. So now coming back to the question. So if anion is negative charge, so that means in anion exchange chromatography we are purifying the anion, right? So in that case, what will be the column that will be packed with? So if we are separating anion, okay, so that means these are the negative molecules that needs to be purified. So the base that we have must be positively charged. That that's how the negative charge will come and bind to this positive charge, and when while purifying we'll take out this negative charge from the column. Okay, so in that case, this is a anion exchange chromatography. That means the anion, that is the negatively charged protein or the negatively charged n halide, needs to be purified from this particular column. Okay, so the stationary phase is positive charge. So this is anion exchange chromatography. So the correct answer here is option number 1. Now the reverse will happen in cation exchange chromatography. In that case, we have a negatively charged beads that will attract the positively charged analyte, and that will be eluted out after that. Okay, so am I clear? And this question clear?
Yes, sir.
Okay, so let's move on to the next question. So DAE Cephardex column is used for purification of basic proteins, acidic proteins, acidic and basic proteins, or acidic and neutral proteins. Can anybody guess? So first of all, DEAE stands for diethyl aminoethyl. Okay, so if anytime amino comes in any of the beads, that means it's a positively charged matrix or positively charged beads. So it will attract negative charged molecules. So it's a anion exchanger basically. Okay, so this anion exchanger will help in extracting proteins that are negatively charged, and here we'll be using it for the purification of acidic proteins basically. Okay, because here the acidic proteins will have constituents like sulfate, phosphate. These kind of molecules will be present on the acidic proteins. That's why these are negatively charged. So here DAE column will be used to separate these acidic proteins. So is the question clear?
Yes, sir.
Okay. So let's move on to the next question. Which of the following techniques is best suited for initial purification of untagged protein from a mixture of various plant proteins? Is it gel filtration chromatography, ion exchange and hydrophobic interaction both, only hydrophobic interaction chromatography, or affinity chromatography? So what can be the correct answer? Guess. So any guesses?
Sir, two.
Okay. Any more answers? So whenever we are extracting any protein or purifying any protein, so in that case, so from a mixture of proteins, for example, here we have the leaf or any of the organs that you consider in plant. So here, from here we isolated all the crude extract of proteins. Now you have to separate those proteins, or isolate a particular protein that is present in this crude extract. Okay, so what we'll do, any purification or any like uh, chromatography that we use, we'll use multiple chromatographies to separate a particular or purify a particular protein. Okay, so one chromatography will not be used for purification at all, because the protein itself has multiple properties. For example, it has charge, it has different size, it has different hydrophobicity. So combination of multiple chromatographies would be used, and here it's an untagged protein. So in case of affinity chromatography, we generally use a tagged protein, okay, to purify it from a crude extract. So the affinity chromatography is not involved here. Now, we don't know any either of these things. Okay, we might know the size based upon the gene length or something, but we might not know the size and hydrophobicity of the particular protein. So now we'll use a combination or both. Okay, both ion exchange as well as hydrophobic interaction chromatography for purifying the particular protein in the initial stages itself. Then further we can apply the isoelectric point wala concept and different concepts to study that particular protein. So here the correct answer is both ion exchange as well as hydrophobic interaction chromatography. Okay. So any doubts in this particular question?
No, sir.
Okay. If not, then we'll move on to the next question. So, given below are the steps for preparing a matrix beads for an ion exchange chromatography. So, removal of these are the steps, and we have to make it in a correct order. So, the first is removal of very small particles, then equilibration of with counter ions, and then swelling of medium. So, what can be the correct order for these particular steps in ion exchange chromatography?
Sir, two.
Okay. Any more answers? So, what generally we do is first for uh, preparing matrix beads basically. So we pack the column with the beads, and then we allow the beads to swell. So this is the first step that basically activates the charge group on the beads. So this is leads to activation of charge groups. Then comes the removal of the small particles. Okay, because there can be some molecules that can go and bind to these particular charge groups that would lead to improper binding. Okay, so what we do removal of very small particles. Then the last step, what we do, we add the counter ions. Okay, so what are counter ions basically? For example, if you have to separate a negatively charged proteins and you have a positive charge on the beads, so we cannot keep this column as it is. Okay, so what we have is we have counter ions like small, these are small ions like chloride ions that can go and bind to these positively charged beads, and whenever the protein comes that is also a negative charge, that will replace this positive replace these counter ions with the protein, and this protein will go and bind to this particular uh, molecular bead. Okay, so this is the role of counter ions. They basically help in binding of those particular proteins that needs to be purified. Okay, so the last step is equilibration with the counter ions. So the correct order would be 3, then 1, then 2. Okay, so the correct answer is option number 4. So any doubts in the steps?
No, sir.
Okay, so if not, then we'll move on to the next question. The interaction of proteins with the HIC, that is hydrophobic interaction column, involves which of the following interactions? Ionic, hydrophobic, covalent, or hydrogen bonding? So yes, as the name suggests, the hydrophobic interaction chromatography requires hydrophobic interactions for the particular proteins to bind to the column. Okay, so the correct answer is option number 2. Moving on to the next question. Which type of chromatography can be used for purifying heavy metal contaminated water? Gel filtration, ion exchange, hydrophobic, or affinity chromatography? What can be the answer? So, whenever we have to purify water, okay, that is contaminated with metals, so in that case, metals are always considered as a positive charge molecule. Okay, so in that case, if we have to separate or purify, or in scientific terms, what we call it as softening of the water. Okay, so in that case, we use ion exchange chromatography to purify the water from contaminated with heavy metals. Okay, so in that case, the correct answer is option number 2, and here is the explanation for that. So in that case, the hard water that we see is contaminated with calcium and magnesium. Okay, that leads to lathering of soap and leading to wastage of the soap particles basically. Okay, so in that case, what we do is we separate the ions with the ion exchange column that have negative charge beads. Okay, that is a cation exchanger basically. So here we'll pass the water that have the contamination of calcium and magnesium to the water, to the beads that can replace these counter ions and will lead to purified water that will have only sodium. Okay, so this is the case where softening of the water comes into picture. Okay, so here we'll be using an exchange chromatography for the same. So any doubts in this question? If not, then we'll move on to the next question. The HIC column, in the matrix with aromatic ring makes additional dash interactions. Cation pi, pi pi, polar pi, or anion pi. What are the interactions that are involved in a matrix with aromatic ring in hydrophobic interaction chromatography?
One, sir. Cation.
Cation
Yeah.
pi. Okay. Any more answers? Okay. Any more answers? So, can anybody explain what is a aromatic ring? Just like the basic. I don't want the specific chemical formula or something. Just the basic. Yeah, sure. So the aromatic ring is like benzene. Okay. So this is a typical benzene structure. Okay. And what happens is, because this has a double bond structure like this, and it also has a resonating structure. Okay, so this is a resonating structure. That means the double bonds that we have will be uh, separated out while after every resonance. Okay, so in this case, the benzene ring particularly will have pi character. Okay, that because of the double bond that is present here. Okay, I'm not going into the specific chemistry kind of stuff, because it's out of the concept of this course. And here, if any time we consider as a double bond, okay, so in that case pi character comes into picture. And in case of aromatic rings, the pi character is the highest. Okay, and these aromatic rings are hydrophobic in nature. Okay, so that is why whenever we are using some organic solvents, so these are water hating, so they do not dissolve in water at all. Okay, some of them would do based on the different charge compounds that are attached to those particular compounds, but most of them would be hydrophobic in nature. So in this case, they will have pi character. Okay, and the hydrophobic molecule that are attached to the beads, that will also have some pi character based upon the compound that we use. So these aromatic rings will interact with the hydrophobic molecule based upon the pi pi interactions. Okay, these come under the van der Waals forces basically. Okay, so here, when we are using an aromatic ring, they will bind through the hydrophobic interactions along with the pi pi interactions as well. Okay, so the correct answer is option number 2. So just remember, in case of hydrophobic interaction chromatography, whenever we are, yeah, so whenever we are using any aromatic rings, so in that case, the pi pi interactions will come into picture. Okay, so I'm explaining the question again. So what will happen? We see that there are aromatic rings. Okay, so in case of proteins, whenever we see, we have three basic aromatic amino acids. That is phenylalanine, Okay, so phenylalanine is F, then we have tryptophan, and then we have tyrosine. So these are the aromatic amino acids that are present in the protein. So whenever we allow these proteins to use for separation in hydrophobic interaction chromatography, so we use aromatic ring that would have this kind of structure. Okay, so this is a basic aromatic ring kind of structure, and because of the double bond that is present here, it has pi character. Okay, so pi character is a feature of the double bond because here the sigma electrons. So there are different orbitals involved in formation of the double bonds. So pi orbitals are also involved in the formation of the double bond. That's why whenever double bond will be present in a molecule, it will have some sort of pi character. So peptide bond also have some sort of uh, pi character in it. Okay. Now, when we use these aromatic rings for separation of a particular compound on a hydrophobic interaction chromatography, on that basis, the hydrophobic column would also have some of the molecules that would have some pi character. Okay, those molecules will not be charged at all because the proteins that we are separating out here is based upon their hydrophobic character. Okay, so the molecules that are bound on these particular beads will also have hydrophobic character, and additionally they would have pi character in them. So whenever a protein carrying either the like for example activated aromatic amino acids would be passing through this particular column, so in that case they would they would interact with the hydrophobic interactions as well as with the pi-pi interactions as well between the molecules that are present on the stationary phase and the molecules that are bound on the protein itself. Okay, so that's why the pi-pi character is additional bond that is present if we are making the aromatic ring while a picture into play. So, is the question clear now? Okay. So, let's move forward. How is protein eluted out of the hydrophobic interaction column? By increasing the salt concentration, by increasing the protein concentration, by changing the polarity of the mobile phase, or by changing the pH of the mobile phase. What can be the correct answer? So, in case of a hydrophobic interaction chromatography, just as I said before, there would be hydrophobic groups that are present on the stationary phase. Now, when a protein is bound to these particular groups, in that case, what will happen? The charges on the protein would be masked by the salt concentration because of the salt. Okay, so what will happen? the salt concentration that we put into the solution, so to basically elute or separate out these particular proteins, mask the protein molecules on the surface. Okay, so that the charged molecules on the surface are masked. Now, when we have to separate this particular protein molecule from the column or the stationary phase and make it dissolve in the mobile phase, in that particular case, what will happen? We have to decrease the salt concentration on the surface. That means we have to expose the charged groups that are present on the surface. So in that case, what we do is we generally decrease the salt concentration, or we change the polarity of the mobile phase, so that the salt that is present on the molecule, on the protein molecule, will be decreased, and it will loosen up the interaction between the stationary phase and the protein, so that it will be eluted out. Okay, so in case of eluting the protein in a hydrophobic interaction called chromatography, we change the polarity of the mobile phase. Okay, so this is the correct answer. Anybody has any doubt in this question? So if not, then we'll move on to the next question. So salting in is the process in which salt helps in increasing the solubility of protein. Salt exposes hydrophobic group of proteins. Salt induces protein precipitation, or salt decreases the solubility of the protein. What can be the correct answer? So here the correct answer is salt helps in increasing the solubility of the protein. So how? As I have told just now, the protein has, in normal scenario, for example, it has a water phase as a solution. So in that case, it has a hydration cell, that is water surrounding the protein. So if in that case we have to first remove this water shell or the hydration shell to make it insoluble or soluble inside the particular organic or inorganic solvent. Okay. So now what we do, we start adding the salt. Okay. Based upon the ionic strength of the protein, it first increases the solubility of the particular protein in that particular solvent. Okay, so while increasing the salt concentration, it increases the solubility to a maximum. Okay, now while it is increasing the solubility of the protein, this process is known as salting in, because here after adding the salt, then the protein dissolution is more after a certain extent. Okay, so at this point, when the protein has maximum solubility in the hydrophobic or non-polar solvent, so in that case what happens, the salt has already masked the charged amino acid or the charged components of the surface of the protein. So that will not be able to interact with the organic solvent. Now, when this particular protein needs to be precipitated from that particular organic solvent, then we increase the concentration of the salt. And after the increasing the concentration of the salt, what will happen? The protein will be fully surrounded by the salt molecules. So in that case, what happens? The protein gets precipitated out. Okay, and this process of precipitation after adding more amount of salt, this is called as salting in. Okay, so this process is known as salting in. So salting in is basically dissolving or increasing the solubility of the protein, and salting out leads to decrease in solubility of the protein based upon the salt concentration. Okay, so any doubts in this question? Okay, then move on. Let's move on to the next question. The use of high salt concentration for binding of proteins into the HIC column is because it helps in protein in forming hydrogen bond with the column matrix. It removes small molecular weight proteins that interfere with binding. It removes hydration layer of the protein and exposes their hydrophobic residues. And it precipitates the proteins which stick on the column matrix. What can be the correct answer? Okay. Any more answers? So whenever we are passing any protein through the hydrophobic interaction column, so in that case what happens? First, we add more salt concentration to the protein so that it will be precipitated out. Okay, or specifically it masks the charged groups on the surface of the protein so that the hydrophobic portion of the column can interact with the non-polar or hydrophobic portion of the protein so that they can make a so that they can interact basically. Okay, now when we have to elute out these proteins, then we start decreasing the salt concentration or change the polarity of the solvent or the mobile phase so that the interaction between the protein and the stationary phase is loosened up, and the protein that we need to elute out can be eluted out in the mobile phase. Okay, so the salt concentration basically helps in removing the hydration layer that is surrounding the protein, as well as masking the charged components of the protein surface such that it can interact with the surrounding hydrophobic beads. Okay, so the correct answer is option number 3. So any doubts in this question? If not, then we'll move on to the next question. Sodium azide is used in column chromatography as it helps to prevent microbial growth while storing the column, remove non-specifically bound proteins, wash the columns with after elution, or equilibrate the column. What can be the correct answer? So sodium azide is antimicrobial in nature. Okay, so it helps in preventing the microbial growth while storing the column. Okay, so this is the main function of sodium azide. So the correct answer is option number one. Okay, so let's move on to the next question. Which of the following is a type of adsorption chromatography? Is it affinity, gas liquid, paper, or thin layer chromatography? Okay, affinity chromatography. Other answers? Okay, any more answers? So, in case of affinity chromatography, what we do is we generally, so in case of affinity chromatography, the protein molecule or the analyte is basically tagged with the molecule so that this particular tag will bind to the receptor present on the beads. Okay, so this is also absorbing a particular protein on the beads itself. Okay, so this is a type of adsorption chromatography. Then gas liquid chromatography is based upon the dissolution of a particular protein on the medium. So this is not an example of the absorption chromatography. Then paper chromatography is also a part of the like where different like in starting paper chromatography was only used for separating the molecules. So this is also a type of adsorption chromatography itself. Okay, but basically it will come into absorption. And thin layer chromatography is also based upon the adsorption chromatography. Okay, because the layer that we use here, the molecules basically adhere to the stationary phase. Okay, before moving on to the stationary phase. So here the correct answer would be 1 and 4 both. So 4 will also be the correct answer there. Okay, so let's move on to the next question. Which of the following is best suited for purifying native proteins from the muscle cell? Can anybody guess? So the correct answer is blue sapphirose chromatography. So what happens specifically in case of muscle proteins? These are large proteins, okay, that are involved in the strength of the cell walls, cell membranes of the muscle cells. So in that case, we have to use blue sapphirose chromatography. This also lies in the range of ion exchange chromatography, where charge as well as the size of the beads also play a role in separating the proteins. Okay, so this is the correct answer for separating the proteins that are basically present in muscle cells. Okay, so let's move on to the next question. In hydrophobic interaction chromatography, the salt concentration of the equilibration buffer should be above the salting out effect. Specific interaction between substrate and enzyme occurs. The hydrophobic patches on the analyte interacts with the hydrophobic groups on the matrix, or hydrophobic groups present on the matrix interact with the charge group of the target proteins. What can be the correct answer? So yes, as I told you before also, while adding salt on the particular protein, it masks the charged molecules on the surface of the protein and exposes the hydrophobic patches present on the surface of the protein. So these hydrophobic patches go and interact with the beads that are hydrophobic in nature or that have non-polar groups. Okay, okay, wait. Can we do it in the last, if possible? Okay. Just remind me before closing up. Okay. And so what happens after adding salt to a protein? It masks the charged molecules on the surface of the protein. Now, what happens? Uh, this exposes the hydrophobic patches present on the surface of the protein. These hydrophobic patches will go and interact with the hydrophobic molecules that are present on the beads, okay, that are non-polar in nature. So these interact and form hydrophobic interactions. Okay, so in hydrophobic interaction chromatography, these are the major key players in separating the proteins. Okay, so the correct option is answer number 3. So am I clear with this question? Okay, so let's move on to the next question. A protein has a pi, that is isoelectric point of 4. At what pH it can be purified using cation exchange chromatography? Now here comes the concept of pH and pi relation that I taught you in the last class. Okay, so someone said 3. Any more answers? So what happens? when we study these kind of questions. So first, what we have to check, what is cation. In this case, it will be positively charged. Okay, so we have to make the protein positively charged so that we can use cation exchange chromatography for that. Now, at which pH the protein will be positively charged. Okay, so then we have to remember the concept of pH and pL. So at which pH, or like the pi at which pH the protein will be positively charged, less than pi or more than pi. Yes. So when pH is less than pi, then it will be positively charged. And the other case scenario, when pH is greater than pi, in that case, the protein will be negatively charged. So here we have to choose a pH that is less than the pi so that we can use the cation exchange chromatography. Okay, so the correct answer is option number 3, because 3 is less than 4. So in that case, the protein will be positively charged. Okay. So the correct answer is option number 3. Any doubts in this question? Okay. So if not, then answer this question. This is also a related question to the last question. So there are four proteins A, B, C, D, and the PI is given for each protein. To purify protein D from a mixture of proteins in a single step using ion exchange chromatography, what combination of buffer pH and resin combination would you select? Okay, so this thing, and let me know the answer. Okay, any more answers? So as we studied last time in the last question itself, if we have to separate protein D that has a pi of 9, so if we have to make it a positive charge protein, then we have pH that is less than 9, And if we have to make it a negatively charged protein, then we have pH that is greater than 9. Okay, so now we'll see the options. If we have a pH 11, that means the protein will be negatively charged. Okay, so if that is a negatively charged protein, then we'll be able to use anion exchange chromatography, okay, or anion exchanger resin. So the first option is wrong. Okay, in the second option, if pH is 2, that means it is less than pi. So this makes the protein a positively charged. So here we require cation exchanger. Okay, so this option is also wrong. In case of third option, the pH is 6, that is less than 9. Okay, and here also the protein will be positively charged, and here also it will require cation exchange. Okay, so this option is also wrong. The last option, in which pH is less than pH is 8, that is less than 9. Okay, so here the protein is positively charged, and it will require cation exchanger resin for the protein to elute out. Okay, so the correct answer is option number 4. Okay, so any doubts in this question? So if not, then we'll close the session for today. Oh, sorry, there is one more question. Anion exchange chromatographic methods at pH greater than 12 with sodium hydroxide or sodium hydroxide slash sodium acetate solution as mobile phase is applied usually for separation of anions, sugar, proteins, or carbohydrates. What can be the correct answer? Okay. Any more answers? Just read the question twice first. Here we have specifically mentioned that pH would be greater than 12. Okay. So if we are separating anions using an anion exchange chromatography, then pH would not matter much. Okay. In case of proteins, it would matter, but in case of other biomolecules, in case of other ions, it would not matter. So here, anions is not the answer. Okay. The correct answer is carbohydrates. Okay. How? Carbohydrates have which functional group? These are basically aldehyde or ketone groups, right? This is the functional group present in the carbohydrates. And then they also have various alcohol groups. So now what we do is, at pH that is greater than 12, what will happen? This alcohol group will turn negatively charged. It releases the H plus ions that are present on the molecule at very high pH. Okay, so this leads to developing a negative charge, and this negative charge on the carbohydrate molecule helps in separating out in an anion exchange. Okay, so is the explanation clear for this particular question? Okay, so this was the last question for today. And if you guys have any doubt in this particular presentation or the uh, discussion that has happened in the course, you can ask. Yes. So first we'll discuss that question. Yeah, this one, right? Okay. So what happens in case of muscle cells? We have proteins that are large, as well as they are like, we have to separate them in like we cannot tag them basically, okay, because of their huge size, and they are involved in the strength of the cell membrane or the muscle membrane. Okay, so for that, what we do is we have to separate them on the basis of their charge present on the molecules. So what we do, we have a specific kind of column that is blue saccharose that requires a dye that is blue in nature. Okay, so that dye name is basically just a second. Uh, so that dye name is Sibacron blue. Okay, and it is a positively charged dye. Like Comasic brilliant blue, it is also a positively charged dye and help in binding to the negative charge protein. So, and the proteins that are present on the cell membrane are mostly like nearly negatively charged. So, the muscle cells or the proteins that are present in the muscle cells are basically used to purify proteins using this particular chromatography specifically to get a pure uh, concentration of those particular proteins. So, am I clear? Okay. So, any more doubts in this particular presentation or in the course content? So, if not, then we'll close the session for today. So, thank you guys for joining.